For 2 decades, 3 distinct literatures in neurobiology have operated in near-total isolation. One group studies reelin, a protein that suppresses toxic tau. Another maps the perineuronal net, a physical shield wrapping cortical neurons. A third tracks the locus coeruleus, a tiny brainstem nucleus that regulates arousal. These 3 fields have been describing the same pathogenic cascade in 3 different vocabularies, without recognizing they are looking at the 2 ends and the middle of a single cut. Conventional accounts of Alzheimer's disease treat the destruction of the brain's extracellular matrix as an anonymous, late-stage event. In this view, amyloid plaques accumulate, microglia activate, and the resulting inflammation indiscriminately digests the surrounding neuronal nets. The clinical timeline suggests a different trajectory. The degradation of the protective matrix follows a specific chronologically ordered demolition sequence initiated by the earliest known pathological lesion in the brain. The command to execute this demolition originates entirely outside the cortex. It comes from the locus coeruleus, a small cluster of neurons in the brainstem. As this structure begins to fail, it broadcasts a dysregulated, amplified signal along its massive network, flooding the cortical layers above with the neurotransmitter norepinephrine. When that noradrenergic signal arrives at the cortex, it commands local support cells to manufacture and release a specific tissue-cutting enzyme, matrix-metalloproteinase 9, or MMP9, to execute the demolition. The loss of the cortex's defense against tau tangles follows a mechanical dismantling operated by the brain's own dysregulated arousal center. The locus coeruleus marks the earliest physical site of Alzheimer's disease due to a native architectural flaw. Unlike the neurons it projects to, it lacks an aggrecan-based perineuronal net. It possesses no structural shield. Because its massive membrane surface is entirely exposed, it is the first structure to internalize hyperphosphorylated tau. Soluble pretangles actually form inside these brainstem neurons in young adulthood, decades before the onset of clinical dementia. As the locus coeruleus accumulates this pathology, the neurons enter a protracted phase of compensatory hyperactivity rather than immediately falling silent. This hyperactivity causes the nucleus to broadcast a continuous, deranged excess of norepinephrine across its entire forebrain arborization. Simultaneously, the receiving synapses in the cortex upregulate their own adrenergic receptors. The brain attempts to catch a falling signal, which amplifies the deranged broadcast during the preclinical window. The ultimate depletion of norepinephrine is an endgame. During the long interval in which the disease is actually decided, the cortex remains exposed to years of toxic, amplified noradrenergic drive. To see what that drive destroys, we first have to look at how a healthy cortical neuron defends itself. This defense relies on reelin, a secreted glycoprotein that functions as a continuous cell-autonomous brake on tau hyperphosphorylation. Reelin requires a specific multi-part staging ground built into the extracellular matrix at the neuronal surface to function. The first components of this staging ground are the lipoprotein receptors, ApoE2 and VLDLR, which protrude from the cell membrane. Binding to these receptors in isolation fails to trigger the signal. An obligate co-receptor is required, N-sulfated heparin sulfate. This sugar clusters the receptors to receive reelin. The signal then crosses the membrane, phosphorylating the adapter protein DAB1 inside the cell. DAB1 directly suppresses glycogen synthase kinase 3 beta, the specific kinase responsible for hyperphosphorylating tau and driving it into tangles. The internal inhibition of tau tangles depends entirely on the continuous structural integrity of this sulfated multi-part surface complex. This is the point where the dysregulated signal from the failing locus coeruleus arrives at the cortical synapse. Excess norepinephrine binds to beta-adrenergic receptors on local microglial and astrocytic cells. This binding triggers an intracellular cyclic AMP and protein kinase A pathway. These signals mobilize transcription factors that move into the nucleus to alter the cell's output. The glia manufacture and secrete the enzyme matrix-metalloproteinase 9 into the extracellular space. MMP9 is a gelatinase that targets the reelin staging ground. This release of MMP9 is a specific mechanical response dictated by the failure of the brainstem's arousal network. When activated, MMP9 arrives, it begins the demolition. It cleaves the lectican core proteins holding the net together. Next, it shears the heparin sulfate co-receptors, breaking the receptor cluster. Finally, it strips the lipoprotein receptors off the surface, silencing the reelin signal. The reelin molecule remains intact, but it is now useless because its receiver has been dismantled. Simultaneously, the same excess norepinephrine that called up the MMP9 shears enters the neuron to directly overactivate the GSK3 beta kinase. This creates a pincer effect. The enzyme cuts the reelin brake, while the norepinephrine floors the kinase throttle. Through this dual mechanism, the locus coeruleus coordinates the total release of the tau kinase from both inside and outside the cell. This mechanical unstaging explains the rising reelin paradox observed in the cerebrospinal fluid of Alzheimer's patients. This chart shows the relationship between total reelin protein and its signal output. Reelin levels climb steadily as the disease progresses, but DAB1 phosphorylation collapses. The signal fails because the receiving surface is destroyed, despite the presence of the protective ligand. Soluble fragments of the ApoE2 receptor float in patient spinal fluid as the physical wreckage of MMP9's destructive shedding. Grounding this framework requires a strict division between established facts and necessary inferences. The induction of MMP9 by norepinephrine is proven in peripheral macrophages. Proving this induction in brain-resident macroglia in situ remains the critical test for this model. Although researchers are still confirming the final glial link, the patient biomarker data reflects a mechanical unstaging of the reelin axis. These mechanics suggest that simply blocking norepinephrine or MMP9 could halt the progression of the disease. But MMP9 is essential for the expansion of dendritic spines during long-term potentiation. To abolish the enzyme is to abolish the mechanics of memory consolidation. MMP9 also helps clear amyloid beta, while norepinephrine is required for arousal and attention. This creates a rigid therapeutic constraint. Abolishing the shears or paralyzing the hand risks destroying memory and clearance systems to save the matrix. Effective intervention requires discipline rather than eradication. The strategy must focus on restoring the natural inhibitor, TIMP1, during the early prodromal window. This would enforce control over the enzyme without silencing its necessary functions. The goal is to reach the locus coeruleus while its signal is merely deranged, stopping the pathological cutting while keeping the weave whole long enough for reelin to maintain its guard.